I was really impressed by the many materials and also techniques that were shown during the last 2.5 days. And I'm really sure that the future of the AFM looks very bright. So what I would add to this is 2 examples from our group where we show how AFM and fluorescence can be used to tackle maybe some additional challenges. So one is, one is the different scales we experience. Yeah, for example, if we look at cartilage, yeah, we heard a little bit about this, where we are on the millimeter or even centimeter scale, down to single proteins, where we are on the nanometer scale. And so I will start with this because you got already a very nice introduction from Matthias Tchaikovsky this morning. And so I want to look at cartilage a little bit from a materials point of view. So it's a hierarchical material. Yeah, it has organization on many length scales. It contains different materials. We heard a lot about collagen. There are proteoglycans, water in there, then different organizations and different layers. And also, in addition, then a lubricant when the cartilage meets the other cartilage. Yeah. And so I want to look at this part here and the lubrication of this part of the cartilage. And several years ago, we are approached from the university hospital at the Technical University Munich, so the Rechts der ISA it's called, from Rainer Burkhardt. And he asked us, I mean, why is synovial, so this kind of resembles the liquid we have in our knee, better than just a physiological sodium chloride solution or even water, if you put this into the patient's knee after a surgery? I mean, you always lose then a little bit of liquid and then they put thing in there. And I mean, the patients that got synovial, they say they feel better than the patients that got just a physiological salt concentration. And at that time, people thought the reason is that this is a better lubricant, synovial, yeah, that the friction coefficient is lower. And so what we used is friction force microscopy, which was also nicely introduced during the last days already. And so we just used this to get a friction coefficient. And what you can see here is that synovial is not better than the others. Yeah, it even looks as if water is a better lubricant here. But let's say this is all not a very good lubrication, actually. But if you think about this afterwards, you can say, okay, maybe the knee is not optimized for being ideally lubricated, but maybe it's optimized for something else. For example, wear. Yeah, I mean, you want to use your knee for many tens of years. So therefore, it would be good if there wouldn't be so much wear. And this was the next thing that we looked at. And so this is a collagen network. So this is what you have seen from Matthias Tchaikovsky, the healthy cartilage network. And so what we did is we used cartilage samples from sheep, it was at this time, so 2015, and used a tribometer and a bead, and really went for 1 hour on a cartilage sample under these 4 different solvents. And what you see is that with synovial, it really looks still more or less like you can nicely see the collagen fibers, and there's not much debris on this, and there's also not much of this, yeah, we don't know, really clumped fibers or so that come together. Yeah, so if you have sodium chloride, there's already more of this, and then if you have just pure water, it's really blurry and the surface doesn't look very well. So the conclusion there was that we have less wear with synovial, and therefore probably our cartilage is optimized for little wear. So here in Freiburg, we were then came together with Bernd Rohlaufs from Gern, the Freiburg University Medical Center, and he's interested in osteoarthritis. So you already also got an introduction into this from Matthäus. Just want to mention there that it's really a chronic disease. Many people, probably even from us, will suffer if they get very old. And the question is, how does this come? What is the reason for this? And so here, Bernd Wolofs, he came up with something on a more almost millimeter scale. So he looked at the organization of the chondrocytes. So these are the cells in this cartilage sample. And this is the one slide you already saw in Matthäus' talk. So he categorized this organization of the cells in different patterns. And this is basically healthy one, and then earlier osteoarthritis, and so on. And what was known before, that also probably this affects the elastic modulus. And this is what Matthäus showed you in detail, that we could show that this correlates, the pattern of the cells and the elastic modulus. But what comes in addition here now is that we really can now connect the scales in one sample. So this is a more than 2 millimeter in diameter sample. You see at the same time, you see the fluorescence, so you can zoom in. So you see the fluorescence of the stained cells here, in this case, palladium iodide. And you see then the patterns, here a string, here a double string. Then you can do force maps on all these positions, 108 positions this was, and you can get then the microscale elastic modulus on all these positions. And you see that the organization is basically higher here, so better. Cartilage here, it gets less. And you also see that the elastic modulus decreases here. And I have to say, so if you just look at this completely macroscopically with the light microscope, it looks intact. So this is basically, this is after surgery of a person who had a problem with a knee, but this is basically the best part you could extract from this, and it looked intact. But here you see, there's already something going on. And now we want to look at the even smaller scale, that is on the nanoscale. And this is what you nowadays can do. And so here we use the QI mode. It's similar to FFM mode or pinpoint mode or peak force mode. So every company calls it a little different. So this is, I think, something like 3 million force traces that we will see now the result of this that Matthäus took. And this is a movie. I will just move a little bit forward and backward here. So this is the sample from before. You see the organization changes from the bottom to the top. Elastic modulus is reduced. And then you see something like a pretty intact collagen network with also some very thick fibers that are important, we believe. And then if you move through the sample and go, you see this on the right here, that you get more and more of these, let's say, clusters here where they clump together, maybe also more of this debris. And then if you go then further and further, it gets more and more blurry. And at the end, basically, you can't even see the collagen fibers anymore. And so this was the same sample as before. And I cannot mention this often enough. So it looks intact, if you look at it by eye. And this is all imaged in buffer. So therefore, I think that I hope that I could show you in this first example that we can correlate now different properties on different scales. So fluorescence for the organization of cells, elastic properties, and the topography for the organization of the collagen network to really not only describe samples, but go on and get mechanistic insights. And now we can really ask the question, okay, what is the reason for this loss in elastic properties, probably the collagen network. And as Matthäus already told you, we are trying now to see and to stain different collagen types to really get at the mechanism of this osteoarthritis. yeah, and as I said, so this was Matthias Tchaikovsky to roll the data, collaborations, Bernd Rolafs, and Bijan Balz. So now I want to jump to the very small size, and just if I have some time, I will tell a little bit about the medium size, but as all of you stuck so well to the time, I also don't want to go over time. And I think this is really something that lies on my heart, and where I think there's a lot of potential that is not yet used in atomic force microscopy. And this kind of structure and dynamics. And what do I mean with this? I mean, you heard a lot about structure determination from cryo-electron microscopy, before it was X-ray scattering, also computer simulations, and now deep learning. And yeah, but more and more people notice now that structure gets easier to get, that dynamics is in principle the key. And that you have to bring these two things together. And I show this now on this one example of the heat shock protein 90. So there's a heat shock protein, but also a molecular chaperone. It's very abundant in the cell. It's involved in many signal transduction pathways. It has several hundred clients, so proteins it acts on. Many of them are kinases. It's a homodimer, and it's an ATPase. But it never works on its own. It always works in complex with other proteins. One, for example, is P53, which is called the guardian of a cell. Yeah, so a very important protein, also involved in cancer. So a lot of studies going on there. And so what we were interested already to start is 10 years ago was the dynamics of this protein. We wanted to see how does it move, and there was this idea that it would make an open-close movement between the two dimers, and with this, and then you would grab a client protein, would catch it, and then process, and then release again. And so we looked at this, and we put fluorophores on there, so 2 fluorophores, and used then single molecule first resonance energy transfer. If you have not heard of this, just think of it as a spectroscopic ruler. Yeah, so if they are far apart from each other, you will see a yellow light, and if they are close together, you will see red light. Yeah, and from this, you can say something about the distance and then about the movement. So this is basically how this would look like. So one of these points is one of the molecules, and we separate the two colors on the two halves of a camera. And what you then have, so you have the one color here, the other color here, and you look at it over time. So this is one molecule. So the one color, the yellow or here green color, and the other one, the red color. That's just a control. We don't have to go into these details. And what you can now from this calculate is something that is called a Fred efficiency, but you can think of this like a distance. Yeah, if this is high, they are close together. If it's low, they are far apart from each other. And so now you can look at the time involvement, so how this moves, open and closes, and you take then much data of this and can then assign the states. I mean, two are easier. You have an open state and a closed state, so open and closed. You see here from the signal directly. Here we see two additional states. You can get them just from the dynamics that is in the traces. For this, several groups developed hidden Markov-based models. So we also have, so this is single molecule analysis of complex kinetic sequences that we developed. And then from, let's say, 100 of those curves, you get then free energy landscape. Yeah, now we are getting closer to the AFM. Yeah, we have also heard a little bit about free energy landscape in AFM. So this is basically what you get from all these rate constants. If you have the rate constants, then you have everything. You have the dynamics, and you have, so to get the kinetics and the thermodynamics. This in principle is the thermodynamic information. And these are the two states you can distinguish, and the two state by the Fred efficiency, and then two by kinetics. And this was done at that time by a graduate student, Sonja Schmidt. So she's now a professor at Wageningen, probably next door to Simone. I don't know, but I didn't have the chance to visit her yet. And what she also looked into is how this is manipulated. So this free energy landscape by other things. And what do I mean by other things? So this was known before that certain amino acids in this HSP90 are very important. And so we mutated them and just looked at how does this affect this open-close movement. And it's very interesting because, I mean, if we do a mutation here, it has a big effect even here on the molecule. And it changes this free energy landscape. So here we collapse it just on one coordinate for simplicity, but in principle, you get all 4 states. Then you can add co-chaperones. So these are helper proteins of this helper protein. You can say, yeah, it's always big protein complexes. And you also see that this free energy landscape is changed. Or you can just add artificially a crowder, yeah, which actually might mimic the situation in a cell where you have many other molecules. And you can then also see that this also affects this dynamics. So basically, if you only had the structure, you wouldn't get this information. But on the other hand, and that's what I want to point out with the 2-point kinetics here, we don't really get information about the structure. I mean, we are kind of just putting these pictures up here, yeah, how this looks like, but we don't know this. Yeah, we just know how 2 points move. So it would be great if we could relate this to a structure. And during the last years, quite some helper tools were developed for this. So one is some AFM viewer simulations that basically, if you have a crystal structure, so a PDB structure, for example, of your molecule, then you can just put this in there and it tells you how this would look like in your AFM image if you put in the tip radius. And that's kind of what we did here. So this is on the one side, yeah, how can you get from structures to the AFM images? Then you can take the AFM images with better and better resolution, and especially with higher velocity. And therefore, you can get more and more images. Yeah? And as we also heard sometime, in the image of one of these proteins that is attached to the surface, and I mean, it shouldn't be attached too strongly because otherwise it will denaturate, it will always be a little bit blurry, yeah, because it's a soft thing there. But now you could use many of them maybe and get something better. And this is actually what was developed during the last years for cryo-electron microscopy. Yeah, Relion is one of the softwares. There are several others also.
You have 3 minutes left.
Yeah, and this you can use now, so you can import AFM TIFF images. You can then pick particles. So in this case, my student Tanumoy, we'll see him on the next slide, pick them by hand, but you can in principle also do this automatically. Working on this, you can then extract the particles, and then you can do classification. And yeah, maybe here is a good point to mention this. So this tool is based on Bayesian analysis, yeah, kind of maximum likelihood. And this will be interesting because there are more and more tools also coming out that use deep learning, and actually some that don't even use data anymore. Yeah, they just do data mining. And it will be really interesting. I think at the moment we should follow both paths, and then we will see what finally is better. I think both will have their advantages and disadvantages. And this is kind of from the first try what you get out. So what I should mention, we did not only look at HSP90. I mean, HSP90 by itself, there's a crystal structure and many things are known. But HSP90 together with one of its co-chaperones, actually the one I showed before, this AHA1. And the question is, I mean, how does this structure look like? And what you already see here, that we get different structures, different classes. So this is what comes out of the software. Actually, we'll still have kind of too many classes. We think, I just put here the first 3 where there are several molecules in these classes. And so you can then, we put the HSP90 in here. As I said, for the AHA1, we don't have the structure yet. But you can think maybe that it's here is kind of then lying here. Here it would be more on the top, and here also. So you get then the different structures. And now you can go back to the fluorescence and look at the dynamics and try to see, okay, what these structures, maybe if you have in the end, I should have put a 4th class here. You have to make this maybe more clear. If you have these 4 classes, 4 structures that you could then relate to the dynamics. And so this is still ongoing work, and I'm happy to discuss this. And so the images were taken by Ramin Omidvar, and the analysis done by Tanu Mondo in its collaboration with Rebecca Wild. And so just in the last 30 seconds, I want to say that the AFM is also, I think, a great tool to look at active matter. And this is something we investigate in a cluster of excellence here in Freiburg, which is called LIVMATS, Living, Adaptive, and Energy Autonomous Material Systems. You have to learn this quite a bit to say it. And the question is, could you produce some nucleotide-driven non-equilibrium structures? Like this microtubule, probably everyone has seen this amazing movies, how microtubules polymerize and depolymerize. Sounds so easy, but if you then think about it, how can you do this? It's tricky. And the first step is you need to get a polymerization and depolymerization. And I think we got this, still not with a complete synthetic system, but with a DNA-based system and two enzymes. And what you see here is basically this DNA plasmid, and then you see some polymerization. So it should be on these two single-stranded overhands. And I think you can see this here, here, and here. And then you see the depolymerization here. And so, yeah, call this a beta version of an active molecular system. And as you see, I mean, it's really important to have the AFM here in real time with nanometer resolution under physiological conditions. And I think you can search quite a bit to find another method that can do this. And this was done by Max and Richard in my group and collaboration with the Walter group. Yeah, with this, I thank my group. I think I mentioned everyone along the talk. I thank you for your attention and for staying here in the conference still on a Friday afternoon. And I'm happy to take questions.